Okay, so today we're going to talk about cellular respiration. So I've got drawn up here, this is a giant cell. So pretend that this is the cell membrane right here. So let's label that. This is the cell membrane. And so this cell would stretch way up into the ceiling and way down into the floor and all the way over into the next room. And inside of the cell, we have, this is just cytoplasm right here, and then we have our mitochondrion. Okay, and our mitochondrion is composed of the outer membrane, which you see here, so we're just going to label that outer membrane. And then we have this inner folded membrane, which we're going to call the cristae, C-R-I-S-T-A-E. And then this space that's between the outer membrane and the inner membrane, wait for it, is called the intermembrane space. And that will become important later. Okay, and then you can see here I've drawn this cell in close proximity to a little capillary, one of our tiny little blood cells. So all cells have to have access to nutrients, and inside here you can see little red blood cells moving up and down. All right, so let's pretend it's first thing in the morning, you're starving, you've been sleeping all night, and you eat a bowl of Lucky Charms. So now your bloodstream is going to be chocked full of sugar molecules, and your cells have none. So I'm going to draw a little sugar molecule as a green hexagon, a glucose molecule. Okay, so these little glucose molecules are floating through your bloodstream. Now if you think about it, we've got high concentrations out here and low concentrations in here. So by diffusion alone, these sugar molecules want to go in. However, they're too big to pass through the membrane. So they're going to pass through this little special receptor that's called GLUT4. You don't need to know that, but just FYI. Okay, so if you remember way back from when we talked about it before about membrane transport, if something's passing down its concentration gradient, but it's being passed through a membrane protein, it's called facilitated diffusion. All right, so right here we're going to do facilitated diffusion. Just sort of trying to pull all the things together that we've learned before. All right, so as soon as this glucose comes in, I'm going to open it up into a 6-carbon chain instead of having it as a ring. So here we have this 6-carbon chain. This is glucose. Okay, so the first thing we want to do, remember, to get the energy out of these molecules, we simply have to break the bonds. Okay, so we're going to start by breaking that very first bond right here. But it turns out to break that bond, we need a little bit of energy to get it started. And if you remember, that's called the energy of activation. And if you remember, when we drew our energy graphs, so this is our energy graph, breaking sugar is an exergonic reaction, but it requires just a tiny bit of energy to get it started. And that was that energy of activation that we talked about. All right, so we're going to put in a little bit of energy. And what's cellular energy? Well, cellular energy is in the form of ATP. I'm going to draw it in red. Okay, so we're going to put a little bit of ATP into the reaction. To be specific, we're going to put 4 ATP. You don't have to know the number. I'm sorry, 2 ATP. You don't have to know the number. We're going to put that up there anyway. Okay, so we're going to put in a little bit of ATP to get it started. We're going to break that bond. So if we follow this down, what are the outputs of this? Well, we have these 2 3-carbon sugars that come out, and these are called pyruvate. And there's 2 of them. Okay, but now we've broken a bond. So what comes out of a bond? Well, you get high energy electrons that come out of a bond. Okay, and then you also get a tiny bit of leftover energy that we're going to put into ATP. So you actually get 4 ATP out, 2 ATP in. So you can see that overall this process actually gives you a little bit of extra ATP. Now what do we do with electrons? So these are high energy electrons out in high energy shells, and we want to store them that way. So if you remember, we're going to temporarily store them on our storage molecule, which is NAD, and we're going to tack on an H, and we're going to put those 2 electrons into that bond right there to store them temporarily for later. So that's our first NADH. All right, so we've done this, and this process has a name. So we've just broken a sugar. So we're going to name it sugar breaking or glycolysis. Glycolysis. Okay, so that's the first step of cellular respiration. Now we've gotten that, we've got some energy, we've got these pyruvates. So at this point we're going to take the pyruvates into the mitochondria. So they're going to pass all the way into the middle. Oh, and I forgot to name the middle. The middle of the mitochondria is called the matrix. Okay, so this area in here is the matrix. So these pyruvates are going to pass into the matrix. So here we are right here. So now what we need to do is break the next bond. So we're just going to break another bond. Okay, when we break that bond, what comes out of a broken bond? You should be thinking high energy electrons, and you would be right. So high energy electrons are going to come out, and these high energy electrons, remember they're way out here, we don't want them to fall down, so we're going to store them temporarily in a bond, just like we did over here. So we're going to store them again in the bond between NAD and H. Now notice we have this single carbon now that's broken off, and it's not a carbon all by itself, it actually is a carbon dioxide. It's got oxygens attached to it. So this carbon dioxide, can you see that? See? Okay. So the carbon dioxide can now diffuse out of the membrane, back to the blood cells, back to your lungs, and you blow it out with your next breath. Okay? So now we've got these two carbons. We need to break that final bond, but it turns out this final bond breaking is super complicated. And it takes 10 steps to do it, but we're not going to learn those steps. We're just going to call it a cycle. So over here in the matrix, we have our citric acid cycle. Okay, and we call it a cycle because it starts and ends with the same product. And so there's this 4-carbon sugar sitting here. It's called oxaloacetate. You don't need to know that, but there's a 4-carbon sugar sitting there. We're going to add these 2 carbons to it, break that final bond, and our byproduct is going to be the 4 carbons again, a 4-carbon oxaloacetate. But turns out these 2 carbons, for some reason, cannot make it over to the citric acid cycle by themselves. They're like little children. They will wander around aimlessly unless you sort of direct them to where they need to go. So we have a little babysitter that takes them. It's a coenzyme, and it's the first one that was characterized, so it's coenzyme A. And we abbreviate that with CoA. Okay, so coenzyme A simply grabs these 2 carbons and takes them over to here and drops them off, and then goes and gets more. And we call this guy acetyl-CoA. Acetyl stands for a 2-carbon CoA. Okay? So it drops those 2 carbons off, and then we break that last bond. So coming out of there, we end up with those 2 carbons breaking off one at a time as CO2s. And those also can diffuse out with the rest of them. Okay? But we've now broken a bond, and as you remember, what comes out of a bond? High energy electrons. So again, you get high energy electrons coming out of here and getting temporarily stored on our storage molecule, which we've learned before is NADH. Okay, we also get another molecule, FADH2. It does the same purpose as NADH, so for our purposes, we're going to leave it as NADH. Okay, and you get a tiny bit of extra energy left over. And so we're going to produce an ATP. But just one per turn of the cycle. Now how many times is the cycle going to turn for a glucose molecule? You actually have 2 pyruvates. So this whole process in the mitochondria gets to happen 2 times. So we'll just put that up there to remind us that the mitochondria happens times 2 for the 2 pyruvates. Okay. So we've now broken apart all of our carbons. Let me label this ATP production really quickly. When we produce ATP this way, we are simply taking a phosphate off of something and putting it onto an ADP to make ATP. It's as simple as that, and we call it substrate level phosphorylation. Phosphorylation is adding that P. Substrate level. This is a really long word. Okay, so that's just normal ATP making. It's not clever and fancy like we're going to see in a minute. All right, so let's count up our carbons. Is our glucose molecule now gone? Well, let's count. We lost 1 here, but we do this twice. So we lost 2 there. We lost 1 here and 1 here times 2, so 2 here and 2 here. So there's all 6 of our carbons, and they're gone. They've been breathed out by you as carbon dioxide. So what do we have left? How have we made any energy? We made a tiny bit of ATP here, we made a tiny bit of ATP here, but that's not enough to keep you alive. So if you remember, what have we done? We've stored electrons over and over again. And these are electrons that are way out here. They're high energy electrons. And if you remember from the fluorescent star, we want to bring them down in energy and suck that energy out of them, but we want to do it slowly so we don't, kaboom, explode, right? So we're going to take it down slowly. So the way to do that is we take these electrons from the NADHs, so from this one, please excuse my arrows, from this one, and from this one, all these NADHs converge right here, and they all have these electrons, and they're going to drop the electrons off on this first protein. Now notice, these proteins are embedded in the Christie membrane. So they are right in between this intermembrane space here and the matrix here. Okay, so the NADHs drop off their electrons. Notice I'm drawing them in orange to try to differentiate for you what I'm talking about. Okay, and each of these proteins has an available electron shell, one lower than the one before it. One lower, one lower, one lower. Okay, and so each time the electrons pass, they're falling in energy, right? So if I were to illustrate this, I'm just going to illustrate it over here. These electrons are being passed from protein to protein like this, right? So these electrons are going down, down, just like the fluorescent star. And each time they go down, they give off a little bit of energy. Now, in the fluorescent star, that little bit of energy came off as a photon and glowed. Now, as cool as it would be to glow in the dark, it's completely useless and it's not going to power your body. So instead of letting the electrons fall and letting the energy come off as light, we're going to use that energy to do active transport. So let's think about that for a second. What was active transport? Let me think. So active transport was taking something from where it's not very concentrated and shoving it over to where it is concentrated. Okay, so we're going the wrong direction. And it doesn't do that spontaneously. We have to have energy. So in this case, what we're going to do is take hydrogen ions, which a hydrogen ion is really just a naked proton, right? It's a hydrogen that we've removed the electron. We're going to take hydrogen ions and we're going to shove them into the intermembrane space. So we use that energy as it falls to shove these hydrogen ions into the intermembrane space, okay? And so what we end up with is a situation like this, where we have tons of hydrogen ions in the intermembrane space and none in the matrix. Okay, so we've built up what we call a proton gradient. Now, this is a whole bunch of potential energy right there, right? Because it wants to go back in. And so we have a special transport protein right here. It has a name. It's called ATP synthase. And from the name, you can probably guess exactly what it does. It makes ATP, but it actually does it through a physical force, which is like the coolest protein on Earth. Okay, so as the hydrogens flow back through this protein, back into the matrix, this protein begins to spin around in a circle. We've actually seen electron microscope pictures of it. I can't even talk. And it starts spinning around. And as it spins around, it actually takes an ADP and a P and it slaps them together as it goes around. So it's using that energy of the hydrogens flowing through it, the hydrogen ions flowing through it, to build ATP. Okay? So out of this comes tons and tons of ATP on the magnitude of like 32 to 34 ATP. So way more ATP than's produced in any of these other ones. And we call this type of ATP production oxidative. Oxidative phosphorylation. Okay, and it produces way more ATP for the sugar, so it's way more bang for the buck. So you want to do this, but notice this key word right here, oxidative. Where is oxygen in this? Well, we haven't written it there yet. So let's go back. Let me use a different color. Let's go back to our red blood cells, and you've got oxygen. It's being transported on your red blood cells, right? and oxygen will diffuse through the membrane. Now, where's oxygen going to go? Well, it's really electronegative, so it's going to go to where it can steal an electron the easiest, which happens to be right over here at the last step of the electron transport chain. So, oh, by the way, this is called the electron transport chain, or ETC for short, electron transport chain. Okay, and so when the oxygen comes here, it steals the electron from this bottom guy. So if you take an oxygen and you add 2 electrons, it's now got a -2 charge, and it's attracted to these hydrogen ions, and so it actually becomes water. Okay, so you actually build water out of the oxygen you breathe in. Think about that for a minute. Where does water come from? It comes from the air that you breathe in. So the CO2 that you breathe out, that's not just the oxygen going in and then coming back out? No. Where does the CO2 come from? It comes from breaking the sugar. So the Lucky Charms become the CO2 you exhale. The oxygen you inhale becomes water. Trippy, right? Okay, so that whole process, once this oxygen steals the electron from here, he steals it from here, who steals it from here, who steals it from here, who steals it from the NADH. So it's really an oxygen's like a vacuum sitting at the bottom of the electron transfer chain, just sucking those electrons down. And so it causes this whole process to run. That's why we call it oxidative phosphorylation. And it produces a ton of ATP. Okay? So there's 3 steps. We have glycolysis, we have the citric acid cycle, and we have the electron transport chain. All right.